Thank you. So, last time we talked, we looked at transport in and out of a cell. We just kind of glanced on things like passive and active transport, and then what your book refers to as bulk transport, which is endo and exocytosis. Today, and then last class, we also looked at some basics about membrane structure. We talked about phospholipids and cholesterol, and then we stopped here. So what I want to talk to you about today is a third major component of cell membranes. And then we're actually going to go back and look at each type of transport again, and in some cases go into a bit more depth, because there are things in, say, passive transport like osmosis that is incredibly important for the cell that your reading talked about, but I didn't even use that word, so we'll go back and look at that in more detail. So again, and this is one thing I like about that Inner Life of the Cell video, sometimes in science we have a habit of oversimplifying. I mean, a membrane is a fantastically complex structure. The whole cell is fantastically complex, and that's what we spend this whole semester talking about, in all honesty. So, I don't want you to think that the membrane is just a layer. It is the outermost layer of animal cells, good. Cell walls are the outermost layer of plant cells. But while it's a layer, there is a lot going on in this layer. So diagrams like this, I think, are really fun to kind of imagine, and then when you see them kind of animated, like in that YouTube video I shared with y'all, you can see that this is a whole world onto itself. We have our phospholipid bilayer. We have little cholesterol molecules that help stabilize the structure, keep it from being too fluid or not fluid enough. But one thing we haven't spent much time on is all these different types of proteins that are embedded or attached to the membrane. So we're going to look at proteins. That's the third major component of a cell membrane, besides phospholipids, cholesterol, and then proteins. Now you can see here that some of these proteins pass all the way through the membrane. Some are found only on the surface, either inside the cell or only on the surface outside of the cell. And some are even linked to this extracellular matrix, which is just this huge network of filaments, things like collagen that we've referenced and that your book talks about. This huge matrix of different fibers that exist outside the cell and between cells. Now, there are a few terms here that you should be noting. One is this integral protein. The term integral protein, and you can kind of see it here, this term integral protein indicates a protein that is in the membrane. It's embedded in the membrane. Some of them pass all the way through. Some of them may not pass from inside to outside, but they are certainly embedded in it. And then the alternative would be what we call peripheral proteins. Just like peripheral vision means the vision kind of off to the side, they are basically attached to one side or the other of the membrane, but not embedded inside it. Now, I'm not going to go through all of these in great detail, and you have this slide and your book does work through it. We will get to some of these things in much more detail in other units. Unit 4 at the end of this semester is all about signals and cell communication. So we're not going to get into much detail here. But you can see here, proteins have a wide variety of functions that they serve for the cell when they are associated with the membrane. We've talked about how they can help things move across, but they can also just be enzymes. And we've talked about enzymes, things that can make or break chemicals down. And some of those enzymes are associated with the membrane itself. Some float freely in the cell, but some of these proteins are enzymes. There are some proteins that help communicate from one cell to the other. Your video project shows a few of those. Proteins that will actually reach out and touch one another. When they do, they create some kind of change, some kind of chemical change that influences things that happen inside the cell or inside other cells. So there is this, they call it signal transduction. That is a fancy pants way of saying you're sending a signal from one cell to another. This one, we don't really get into much this year, but it's phenomenally important. And we will talk about this like when we talk about blood types in the genetics unit. Some of these proteins have special structures on them. What does this prefix glyco in glycoprotein refer to? Sugars. Some of these proteins have polysaccharide chains. And the purpose of them, the purpose of these chains is to basically serve as an ID badge. It's an identification system where cells in your body can look for these badges, these polysaccharides on other cells, and determine, is this cell supposed to be here? Is this me that I'm interacting with? And things like the leukocytes, the white blood cells featured in the video, if they don't recognize a cell, they will kill it. And sometimes usually that's great. That's called your immune system. But sometimes your body messes up and will actually start killing your own cells kind of unintentionally, and that can cause problems. But we'll talk about this cell-to-cell recognition later. We talked about in the last class, or maybe 2 classes ago, about how some proteins actually help lock cells together, like the desmosomes that we talked about a few lectures ago, that help cells kind of anchor one another to each other. And then some proteins actually help anchor a cell to the extracellular matrix. So we have structural kind of communication and identification, and then movement, transport, and even chemical change that these proteins are involved in. Okay, so a lot of this chapter and a good part of this unit is about materials moving across the membrane. How does a cell get stuff in and kick stuff out? And that's what we're going to spend the rest of this chapter on. And all of these things we have touched on, but I'm going to go into more detail with them. So in this case, what you are seeing here is a membrane of some kind, a barrier that lets some things in and doesn't let other things in or allow things to cross. And what you see is a movement across this barrier of particles from higher concentration to lower concentration. All right? And this is an example of what? This is actually not an example of osmosis, and I'll tell you why. I don't mind that guess. These are solute particles, not solvent, but solutes spreading around from high to low is what?
Isolated or simple.
Simple diffusion. Simple diffusion. This is simple diffusion which is an example of passive transport. All right? Now, it's simple and not facilitated because of what? What is not needed in this example?
Oh, transport protein.
You don't need a transport protein in the case of simple diffusion. And it's passive because what are you not using?
Energy.
Energy to make it happen. That's right. So I just wanted to revisit this briefly. This is from your reading. And it's just a nice little diagram of simple diffusion, which we've already talked about. Now, one thing I didn't mention, and some terms that some of you actually referenced in a previous class that I didn't stress, diffusion keeps going really beyond equilibrium, but the goal is to reach what we call equilibrium, where we have what?
Equal on both sides.
A balance in concentration. Equal molarity, you could say, on either side of the membrane. But even when you hit equilibrium, there's still diffusion that takes place. It's just the same amount in both directions. So it's not like particles stop moving just because there's equilibrium. It's just that you have a situation where as many are moving out as are moving in, and there's no net change in where the particles are going. Now, that takes us into, I know it's kind of tiny here, but osmosis. Osmosis. I did not have time to talk about it, and I plan to talk about it later, so here we are. Osmosis is specifically referencing water. Where's the water going? Okay? And you could define it as the diffusion of water. So when you talk about osmosis, we're specifically focused on water. But it's the same principle. Okay? Osmosis happens without energy. It's going to go from high to low. It's just simple diffusion of water. But because water is so important and where water goes is so important, sometimes we focus specifically on it, and that is a term that we use, or osmosis is the term we use for that. So this is a classic diagram that you see all the time in biology, and maybe even in chemistry a little bit when you talk about these things. But here we have this construct where you have a barrier here, a membrane, that will not let solute particles cross. It will only allow the movement of water. And you can see in this diagram, you've got more solute on the right side here versus less solute. So there's a higher concentration in this region. But the solute particles can't cross this membrane. So the only thing we're focused on in an experimental setup like this is where's the water going? And in this case, you are going to see water move from where the concentration of water is higher to where it's lower. Now, this is where some people can get a little bit confused, not really with the concept, but with how we word it, and sometimes the language used in the questions that surround osmosis. And we're going to spend the next slide on this as well, so you'll have a chance to see this in action. But if you're talking about, like, give me the name of a solute. What do you want these green particles to be? What's something we would dissolve in water?
Sugar.
Sugar, great. This is sugar. There's a high concentration of sugar on this side of the barrier, okay? But since there's a lot of sugar, it means what about the water? There's a lower concentration of water. So since you have a lot of sugar here, the water still wants to move from high water to lower water concentration. So a high solute really means kind of a low solvent. So sometimes, it's nice to just be like, oh, high and low. But you gotta say like what's high and what's low.
So the water always wants to, well, obviously the water always wants to go from region of low to high. I mean high to low.
High concentration of water to low concentration of water, which actually means it wants to move to where you have high solute, because that means it's low water. So basically, in this case, if you have a solution that's really sugary or really salty, the water wants to move towards that. And you can even see like this, the water level increases because water moves in this direction towards where you have a lot of solute. But that is simple diffusion. It's just water going to where you don't have as high a proportion of water. Now, you might at this point be thinking like, well, okay, what does it matter? Well, it matters a lot, and you experience this all the time. So in this diagram, and in this part of the book talks about tonicity, which is tonic with an I-T-Y at the end. It's not a term I really use a lot. It's a more general term that refers to these scenarios: hypo, hyper, and isotonic.
Should we write it down?
And I would write these three words down, but we'll start with hypotonic and then work our way to the other ones.
What's the general name again?
So it's tonicity,
Okay.
like tonic, I-T-Y. That's the general term for kind of where water is going and why it's going in that direction. It's really the ability for water to move in and out of a cell, but it's all based, obviously, in osmosis. Now, we have animal cells at the top, specifically red blood cells, and then some plant cell diagram at the bottom. We can really focus more on the animal cell than on the plant cell, but the same thing is happening in both. So a cell can find itself in one of three environments. It can find itself in a hypotonic, isotonic, or hypertonic environment. And it makes a big difference in the life of the cell and what that cell is going to do. So, hypotonic. Okay? Write down the word hypotonic. Do you know what the prefix hypo means?
What?
Hypo.
I'm thinking of
Too much.
It's actually the opposite.
Too less.
It means below.
the first thing I think of when I think hypo, what is it, like hypoglycemic or something?
Yeah, hypoglycemic means your blood sugar is low. What's a hypodermic needle?
Oh, like a very.
Cold.
Hypodermic. What's derm?
Dermic is your skin.
It means skin. A hypodermic needle is a needle that goes under your skin, below the layer of skin. That's what you get shots with. Hypothermia means your body temperature is below normal. So hypo means below or low. Okay? That's what hypo means. So we have to ask ourselves, what's low here? Okay? And I'm gonna explain. So hypo means low. So a hypotonic environment is one that has low solute concentration outside the cell. This is a low solute concentration outside the cell.
So does that mean there's like too much water?
Yes! So think about that definition I just gave you. A cell is in a hypotonic environment. That environment has low solute concentrations. So what is high in concentration? The water concentration is high on the outside. So the hypo refers to low solute on the outside, which means you've got a lot of water. There is nothing more hypotonic than pure water. It's got no solute and it's all water. So let me ask you, if you drop a cell, which really most cells cannot control where water goes. Water is a tiny molecule, it goes in and out based on osmosis, nothing the cell can do about it, or very little in some cases. If you put a cell in pure water, where obviously the concentration of water is very high, where's the water going to go?
Inside.
It's going to go into the cell, and it won't stop moving into the cell until what? It can burst the cell, which we use this term, it lyses the cell. So your cells can actually burst open in pure water. People have actually died from this.
When you drink too much water?
You can drink so much water and make your blood so hypotonic that your red blood cells burst and you can't carry oxygen
But
to where you need to get it.
is it drinking water in one sitting or like what's the word? When you do it like multiple days? I don't know how to no, because like
Well,
does this happen if you just drink water at like one sitting?
you're not going to get hurt if you just and this is super rare, by the way, but you're not going to suffer if you just always have your water bottle with you.
Yeah.
You're drinking a lot of water over the course of the day, and then urinating when you need to. The one case I know of was some radio station somewhere about 10 or 20 years ago had a contest where it was how much water could you drink to win, like, I think it was the console called a Wii. Do y'all remember the Wii? And it was hold your Wii for a Wii. And basically it was how much water could you drink without peeing. And the person that won died because they drank so much water that they created a hypotonic scenario with their blood, and they lysed not all of them, but they lysed a lot of red blood cells, and
Did they die right there?
I think so, or they were rushed to the hospital. But again, look, drink your water. This isn't going to happen to you. The only other time I've heard of this, my brother was in med school, read some paper about like some antipsychotic drug that made people really thirsty, and like one guy literally like stuck a garden hose down his throat and turned it on. I mean, that is an extreme scenario. Okay, now isotonic. Iso means, well, what's an isosceles triangle for you geometry folks?
Not equal.
Two sides.
Two sides are equal.
What is it, two? Or is it all three?
No, two sides are equal. And then
Two sides are equal. Iso means equal. Almost messed up my own example. An isosceles triangle, something's equal.
Okay, wait. Sorry, can I go to the hypotonic situation? So will a plant cell not burst?
So a plant cell has a cell wall that will at least help prevent it from bursting for a while. I don't know that a cell wall would ever break under that pressure. Pressure is actually part of this, and this is the one thing that if I might go into. There's one equation, not that we're going to do any math with it, but I might show you an equation next class. If the cell wall will create so much pressure inside because it won't allow it to burst, that you hit equilibrium. And it will stop just coming in even though the concentration might not be at equilibrium. But yeah, plant cells basically get really crisp. This is why they like spray veggies at, Kroger and Publix constantly. They have a little mister thing just to keep the cells turgid, which just basically means packed with water and crispy. Okay, so instead of low solute concentration on the outside of the cell, isotonic environments have the same concentration outside and inside. So what's the water doing?
Going in and out.
It's going in and out at this equilibrium. It's at equilibrium. So you have a normal situation. All cells want to find themselves in, what that cell would consider an isotonic environment.
So when the water inside the cell is equal to the water outside the cell.
The concentration of water is equal inside and outside. Yes. And then finally we have, the one scenario we haven't discussed, which is hypertonic. And what does hyper mean?
Too much.
It means above, okay, as opposed to below, or high as opposed to low. Where have you heard the term hyper? What's that? Yeah, hyperactive is a technical term for someone whose activity level is higher than normal. So yes, maybe a parent told you to stop. We have turned it into slang of just hyper. And in English, a lot of times when you say hyper, you're actually referring to hyperactivity. And you're saying don't be so hyper, or they are so hyper, meaning they have a high level of activity. But it just means high or above. You could be hyperthermic. What would hyperthermia be?
Too hot.
You're overheating. Yeah.
This has I forgot.
Oh, yes. Sorry. I'll get Maria to hit pause on the video when we're done. Now, what is high? The concentration of solute outside the cell.
There's a lower concentration of water.
Which means a low concentration of water. Yes! So hypertonic means high solute concentration outside the cell. So low water. High solute would mean low solvent. So if you think of it as low water outside the cell, where's the water going? Out of the cell, from high inside the cell to low outside the cell. And cells can shrivel up or wilt if it's a plant. When you see a wilted plant, that is a plant that is suffering from a hypertonic environment.
Is this what happens when you make jerky or something?
Yes, when you salt meats, like if you make jerky, which part of making jerky I think is drying it out and getting all the water out of there. But if you salting things can preserve them because you basically create such a hypertonic environment, nothing, including the bacteria that could cause the meat to go bad, can live there. But especially with a Western diet, you can run into this hypertonic scenario in your body pretty often. So let's say it's Saturday night and some friends are coming over. What kind of things are you eating and drinking with your friends? Legal answers only.
Water.
You're drinking water and eating celery sticks? Is that what you're doing when you have friends over?
Maybe like
Pizza.
Pizza,
potato
potato
chips.
chips,
Wings.
wings,
Just think
soft drinks, candy. All of those things are packed with the solute salt and sugar, which is why you like them. They're salty and sugary treats. Well, if you eat a bunch of pizza and some mozzarella sticks, and you're dipping them in some barbecue sauce, which is basically just a paste of sugar and salt, and then you're drinking these sugary drinks to wash it all down, and eating cookies, little sugar cakes, you're going to have a very high solute concentration in your bloodstream, which means a very low water concentration in your bloodstream. And when you go to bed, you will wake up in the middle of the night or the next morning, and how are you going to feel?
Thirsty.
Thirsty. Maybe sick if you ate too much. But usually you would feel very thirsty as a result. Because where did the water go?
To the bloodstream.
Out of your cells and into your bloodstream, and your cells are sending signals, I need more water. Make me isotonic, please. Give me an isotonic environment. You pizza lover. So your cells can actually shrivel, plant cells wilt when they don't get enough water. So in the interest of time, I don't want to run out of time, and I have a few more slides to go. Your book refers to the contractile vacuole that you can see. This is a single cell organism known as a paramecium. Don't worry about the name of it. But some cells, especially single cell organisms that might find themselves in a hypertonic or hypotonic environment, just based on where they live, some cells can actually take water and use kind of pressure to squirt it out of them. So these contractile vacuoles are capable of collecting water if there's too much water in the cell and sending it out of the cell.
So is that like part of the vacuole, like little arms, or is that like the?
These little star-like things, I don't know if that's like streams of water that are headed out of the cell. Of course, it doesn't really make sense here and here. I'm not sure what that's representing in this picture in real life. But that would be your vacuole, a big vesicle that collects and dispenses water out of the cell if necessary.
Practically giving us all the quizzes.
Yeah, a little quiz answering going on around here. Okay, so that was passive transport, simple diffusion. We're actually still on passive transport here, but what do we call it when we have a protein helping? But still from high to low? High to low. High to low. Facilitated diffusion. Facilitate means to be helped or to help, and it's a protein that is going to help, in some cases, get some materials in or out of the cell from high to low. No energy is required. This is passive. But if the material cannot actually pass through the bilayer, it's going to need to go through a protein that will allow for that movement. So sometimes you need a protein that's facilitated diffusion. I hate to move so fast, but we have talked about some of these, and some of these slides are a bit more of just referencing what we just talked about. There's a lot going on here. This is called a sodium-potassium pump that your book spends a little time talking about. This is an incredibly important mechanism in your body. Your nervous system relies on sodium and potassium moving in and out of neurons. Gatorade calls sodium and potassium what?
Is it electrolytes?
Electrolytes, yeah. That's the fancy word Gatorade calls for the salt they put in their salt water drink. Not to minimize it too much, you do need salts if you lose them through sweating. You do not need Gatorade if you're playing your Xbox. Probably not losing a lot of salts doing
It's on my brother,
that. Now, the way this works is you're actually going to have to, or what the cell has to do, is move some of these ions from low to high. What do we call that process? That's active transport. Active transport. You need a protein to do that. And since you're going against the will of the universe and moving from low to high, what else do you need? Energy. ATP needs to be spent in order to do this. And here we have kind of all of those on one slide. Passive, facilitated, and then when you're going from low to high, active. Which again, keep your eye out for things like this in your book. Anytime you see these little ATP signals here, that is energy is being used. It's not effortless. Okay, so I think we're going to finish. On time. There are a few other kind of more specific, this is an example of active transport, but it's kind of a very specific style of active transport that we call a proton pump. We will see this next unit. So later this month and into November, we're going to be talking about these proton pumps. This is obviously the membrane. This is a protein. This protein is using energy in the form of ATP.
Is that an integral?
It's an integral protein that's embedded and kind of through the membrane. And what are the protons that it's referring to when it says proton pump?
In this example, it's
Hydrogen ions. Now, so I know some of you are in chemistry right now, but if you have hydrogen with one proton and one electron, and you get rid of the electron to give it a positive charge, all you're left with is a proton. So this H plus is often referred to as a proton. And some proteins can take these hydrogen ions and pump them out in order to get a positive charge on one side and a more negative charge on the other. So you can create electrical differences in and out of the cell, inside and outside the cell, by moving some of these chemicals. And this is one basis for your nervous system. But it's also a trick that your cells use for other things. Yes. So you have this fascinating kind of cooperation at times in the cell. And again, we'll get into specific examples. I would just, again, in AP biology, a lot of times what you need to do is, if I were just to throw this at you, be able to talk your way through it. Okay? Using facts in biology. But this proton pump can create what we call a gradient, where you make one side high and the other side low. And then you can use some energy to pump one chemical out, and then you will actually, almost like water running over a dam, use the energy from the chemical naturally flowing or diffusing back in to help move other chemicals in.
So the co-transporter isn't an active, like.
So in the case of the co-transporter, it's much more passive. I don't know that it requires no energy, but it's possible to bring in these big sugar molecules with the help of diffusion that is occurring because you made it high to low with a proton pump. This idea of co-transport is bringing something in or out using the energy from something else moving in or out. And again, it's kind of like a dam. I mean, you build a dam on a river, the water comes up high, and then you just let that water flow over a turbine and you make energy. Kind of a similar idea. You're creating some sort of condition that later you can get the payback from. And then finally, thank you for your patience. The last two types of transport we've already talked about. This would be an example of exocytosis, which again we talked about. This is not like the other transport mechanisms. We can actually use in our cells vesicles that fuse with the membrane, because they are membrane as well, membranous. And you can dump massive amounts of chemicals out, or a mass of chemicals out, by making a vesicle that goes to the membrane, fuses with it, and then becomes part of the membrane, and as a result, just releases a set of chemicals. This is not really based on high or low. I don't know where the high or low concentration is. You're making a package and you're sending it. It's going to require energy, but you can get a cluster of chemicals or molecules in or out in this way. If the chemicals are moving out, we call it exocytosis, and if they're being brought in this manner, what do we call it? Endocytosis. So in this case, they have a few different examples of the membrane actually shifting, surrounding some kind of particle and encapsulating it in a membrane. There are a few ways this happens. Your book goes into like phagocytosis and pinocytosis, which is basically making a big vesicle or making a bunch of small ones. Sometimes receptors, little proteins are involved that help signal when these vesicles should be made. The last slide here is, I only threw this in here because this is a real cell, an amoeba, single-celled, very primitive animal-like organism that is wrapping itself around another cell for the purpose of eating it. It's going to bring this cell inside through endocytosis, And basically, it's how this single cell organism would eat something. Woo! That was about 40 minutes, 45 minutes, pushing 50 minutes maybe. We're going to stop the video. The next class is going to maybe involve one quick thing related to hyper and hypotonic. I'm going to show you an equation, but then the rest of the next class will just be about test review. So you can stop the video and you may go.